Okay, good morning, everyone. It's my great pleasure to welcome you all here for our, I think it's already the 6th edition of our 2000 hands-on workshop in micro and nanobiotechnology. The name has been drifting a little bit, but each year we've had a lot of fun. And this year we had a lot of subscribers. It's our biggest cohort so far, by far actually. Usually we were like in the 20s, but we did a bit more advertising or word of mouth. And so I'm very happy to welcome you all together this morning. And so traditionally we've had, the first thing I asked, and I think I'll do that today, also we have a bit more people today, is actually I asked everyone to briefly introduce himself and explain his motivation, or well, basically explain maybe what he's studying and why he came here and what he's expecting from the course. So yeah, this will take a few minutes more this morning, but I think it's a great way so that we get a brief snapshot of who is actually participating in the course, and as you'll see, it's quite diverse, and maybe it's also a first point of contact where you can identify people who are maybe similar interests, and in general, I think it's a good way to introduce one another since we actually will spend an entire week together. And so yeah, feel free to interrupt if you have any questions. I mean, some years we had some very interactive people, some years a bit less. I think it's great if there's people who ask questions, and then you can make sure that everyone is on board. So don't be shy. I mean, we used to have a bit more faculty, so they're usually less shy, but I'm sure you can all rise up to the challenge. Okay, so let's start with the introduction. So I'm David Juncker. I'm the prof in the other building. You will see my lab when you're doing one of the hands-on sections. And so our lab is actually working in this micro nanobiotechnology field. So we have kind of two approaches. On one hand, we see some biomedical challenges. For example, for biomarker discovery, there's big issues. So we try to develop technologies for that. And on the other hand, we also have this more creative approach where we have new ideas about things we can do, and then we develop them. And over the years, we have run this course, and the motivation was, I think, the primary motivation was basically to make people know the microfab, especially a few years back when many people didn't really know about it, especially on the biological side. And then the other goal in general is to promote this interdisciplinary research, which I think has a lot of potential and is very exciting. And with that, I'll give the word to each of you, so you can each briefly introduce yourself and tell us why you're here. Reza.
Hello, my name is Reza. I'm a new faculty in physiology, and I took this course because it was recommended by a colleague of mine. We work on neurons, so we're trying to find ways to steer neurons in one direction or another.
My name is Ahmed. I'm from Electrical and Computer Engineering Department at McGill. I'm working on localized surface plasma resonance biosensors. So I'm here actually to design microfluidic channel, or maybe nanofluidic channel. I'm gonna see how it works.
Great, thank you.
My name is Camille. I'm in the neuroscience program at McGill, and I'm interested in using some of the microfluidic probes to do some neuronal injury.
Thank you.
Hi, my name is Audrey. I'm from the University of Sherbrooke. I'm here through the ISS program. I work on photodetectors for medical imaging devices, positron emission tomography, and my PhD will involve some microfabrication, so I hope to learn some new things there.
Thank you.
Hi, I'm Samuel Farah, and I'm from University of Sherbrooke. And I'm in the same group than Audrey. So I work on detectors based on photodiode. I'm here to discover microfluidics.
Okay, great, thanks a lot.
My name is Alexei. I'm from the Department of Chemistry. We actually work in the centrifugal microfluidic lab, and I'm here to kind of learn different sides of microfluidics than what we're used to in our lab.
I'm Bouchard. I work in microfluidics in polycarbonate. I'm here for different materials.
Hi, my name is Novid. I'm working in McGill Chemistry. We're working on bioactive papers and like diagnostic system and microfluidics. So we're working on more on paper-based products. So just want to know what's going on here.
Thank you.
Hi, my name is Etienne. I'm in the biology department. I actually study ecology, and I'm mostly here as for a curiosity-based approach. Some people are using printing to create habitats for bacteria. So this studies individual organisms.
Thank you.
Hi, I'm Anne-Ryan. I'm working in the physics department at McGill, and I'm working on cantilever biosensors. And doing my PhD work, I was working with microfluidic sensors as well, so I was curious to see what's new and
Thank you.
Well, I'm Ali from Laval University. I'm really interested in microfabrication technology and I work on hydrodynamics of microfluidic devices.
Hi, I'm Fatima from the Electrical Engineering Department. I'm in photonics school. My research is on polymer-based photonic switches, and I'm here because I have some challenges in my fabrication. I need to do some brainstorming and learn more.
I'll have to ask Lino to join us then.
I'm Maria, Faculty of Neurosciences and Environmental Engineering, and we are going to use this technology on finding some biomarkers in traumatic brain injury.
Hello everyone, I'm Amir. I'm from the Chemical Engineering Department. I'm doing optical tweezers interferometry to characterize soft interfaces, and I believe that microfluidics could be a good asset for part of my work.
Hi everyone. My name is Sadiq. I am new staff in microfab clean room downstairs. So I'm here to develop my knowledge in microfab, as I am more IC designer than microfab.
Thank you.
Hello, good morning. My name is Ian Kang from Concordia University. Actually, my background is material science and engineering. And after my master, I have grown my interest in the biomaterials, especially on the magnesium-based biodegradable materials. So now, today I have the chance to learn the nanotechnology. I'm very interested in this kind of fabrication. Nice to meet you.
Okay, thank you.
Hello, my name is Jonathan, and I'm working on cardiac cells. We try to maintain them in vitro. So I'm interested here about the micro patterning of the substrate to induce some orientation or change in the cells.
Hi, my name is Adeline. I'm in chemistry department at the University of Montreal, and one part of my project is on surface with thermosensitive polymers, so I can hear mainly by curiosity.
Okay.
Hi, my name is Kimmy. I'm a master's student in the University of Montreal. My project is to use the scanning tunneling microscopy to characterize the cell assembly molecules on the surface. And I just want, I came here just to use a different type of nanotechnology.
Okay. Thank you.
Hi, my name is Dennis. I'm from the Department of Mechanical Engineering here at McGill, and we study new guidelines for continuum models of living fluids, such as, for example, bacteria solutions. So I'm mainly here to find out about possible applications since our work is based on theory and simulation only.
Okay.
Hi everyone, my name is Khalil. I'm in the Department of Biomedical Engineering in the lab of Dr. Tabrizian. And my project is about integrating dielectric spectroscopy, which is a detection method on microfluidic devices. So I hope through this course I can add to what I already know.
Okay, thanks.
Hi everyone, my name is Hala. I'm a PhD student at the Engineering Physics Department at Polytechnic. I work with optical microcavities for bacterial sensing, and I would like to know more about the microfluidics.
Okay, great.
Hello, I'm Alexander. I'm from the Department of Chemistry here at McGill, and I'm here primarily to learn how microfab works, what I can do here, how to use it in my research.
Okay, great.
My name is Chris Corkery. I'm from the same group that Alex is, and I'm working on biological soft polymer materials to see how they can be applied.
Okay.
My name is Tom. I'm also from chemistry, and our group looks at photoactive polymers and how solid's wet work on wet surfaces, so I'm interested in applying that to microfluidics as well.
Okay.
I'm Alex, also from chemistry, and I work with photo-responsive materials and interested in expanding my skill set.
Hi, I'm Misha, and I'm also from chemistry at McGill. I'm studying the advanced relative for polymer. So I'm just wondering about how my materials
you. So chemists like to self-assemble in the back. Okay, yeah, please.
Hi, I'm Brian. I'm from biochemistry at McGill. My lab studies cystic fibrosis, and I'm interested in whether we can use the microstab to develop a diagnostic system.
Okay, yeah, and then we'll hear a bit about the organizers.
Hi, I'm Camilo. I was the lab instructor to teach you guys how to use the focus study in life.
Hi, my name is Ayo. I'm a PhD student in chemistry. I work on integrated microfluidic systems where the liquids are moved by capillary forces mainly, or liquids to do with benefits in time to bacterial detection. I'll also this morning be talking about soft lithography, which is making microfluidic devices out of PDMS, which is a soft rubbery material. So,
Thank you.
Hi, everybody.
Go ahead.
Okay. I'm Matthias. I'm actually an exchange here from Switzerland, and I will be taking pictures this morning, and otherwise showing you how to use the gradient generator in the lab sessions.
Hi everyone, I'm Grant. I'm the course organizer this year, so I'm the one that's been sending you all those emails. I work with Dr. Juncker, developing a sensor for detecting pathogenic bacteria.
Hi, I'm Veronique. I'm a PhD student in Dr. Juncker's lab. I work with Marianne over here on finding biomarkers for traumatic brain injury patients.
Hi, my name is Vino, and I am a process engineer at the McGill microfab, and I will present to you today an introduction to microfabrication after the implementation.
I'm Philippe. I'm a student in David's lab. I'm working on capillary microfluidics with alternative materials like red. And I'll be talking about the design challenge.
Hi, my name is Mohsen. I'm a postdoc in Dr. Junker's lab and also one of the organizers. And my research is about fiber-based tissue engineering. I'm using micro-nano technology to create small-scale fibers for
Okay, great. Didn't forget anyone? No, I think we did it sort of. Sure. Okay, well, it's great. As you can see, there's a big diversity of people, and that will be reflected a bit in the lectures we'll give you. I mean, we'll have, I'll get that in a few minutes, but we'll have like the lectures to hopefully bring everyone on board at the basic level. But before just I go there, I just wanted to kind of summarize briefly for everyone who is coming today. So 38. I mean, a few participants, the ones from industry, are joining from Abbott Diagnostics. They're not here today, but the rest, so 22 from McGill, 5 others from Montreal, and 5 from Quebec, including some from Université de Québec en Outaouais. That's, I think, quite far, but I'm not sure they're here today. But so you have some people coming quite far, and participants by job studies, so big majority of PhD students, you just heard, a lot of masters, 1 prof and 1 postdoc. We're happy you guys came here too. Thank you. Every year we've had faculty as well participating. I think it has been very fun for them as well. And then finally, participants by area, and as you can see, as we just heard, it's quite broad. I mean, chemistry here is strong interest. We're happy to have you all here. A lot of then electrical engineering, and then it goes from neuroscience, chemical engineering, mechanical engineering, physics, physiology, biochemistry, biology, 1 biomedical engineer. Thanks, and then physics engineering. So you can see we cover really all the range of participants here. And that's, well, I think it's a reflection of what this is about, nanotechnology bringing together all these fields and where we can start to do exciting things together. Okay, so goals of the course. So I'd like you all to learn a bit about micro and nanobiotechnologies, a bit about their potential, of course, but also a bit about their limitations. Then, I think the big part of the course and the big excitement is to get this hands-on experience with both microfabrication, so you'll go to the clean room, the kind of one-to-one coaching and do all the steps for making a wafer. And then you will take this wafer and go to the lab, and you can make copies of it and then use it for software printing and microfluidics. So get this really hands-on experience. I think that's very important when you understand something, to do it yourself. And then basically you can start to think a bit about how micro and nanotechnologies may help and leverage your research. We already heard several of you, they think about using it, they like to use it. So I hope this will really help and bring you to kind of maybe some concrete answers to that. And well, the brainstorming session, that's I think a great opportunity. If you have ideas, we can discuss them as part of a group. And you know, like we see when you have many brains together, you can have really interesting ideas coming in. And then in the end, and that's something we have like had now 1 or 2 years, we have a design challenge. So this year we'll have quite a challenging challenge. And it's also a great way then for you to apply what you have learned and try to see with the challenges, in developing these technologies and the limitations that you have to overcome. And so we'll talk more about the design challenge later. And then I think in the end it's also learn about the other participants, and we already did that. So you can see, I heard like people working on bacteria, one theoretical, one more experimental. I think that's a great way where you can start to talk. And basically we'll learn about other things that maybe you haven't heard about before. Okay, and then the schedule and locations, I think you have seen it. I mean, all of you made it down here. So that's where most of this will take place, both today and on Friday. It will be here. Then the clean room part, that will be in the same building here, but I think it's 1 floor down. It's room 011, so you'll have to go there whenever you are on schedule to go there. And then for the session in the lab, the microfluidics or microcontact printing module, that's in the Innovation Center. That's the Genome building. It's just across the street. I mean, it's closed now, but if you look there, that's just up the hill, the modern building on the side, next to the wall, and that's room 6500. So if you have any questions about where to go, just let us know. Here's actually a map, so you can see it from the top. So we are sitting somewhere here in this building. That's where most of the action is, and then that's the other Innovation Center here. Well, you can come from Dr. Penfield now. There's the construction, so you have to come either from the top or from campus. But I think it's quite straightforward to find it. Okay, so course modules. So we have a module this morning, Introduction to Micro Nanotechnology. So there's 4 lectures and an introduction to the design challenge. And so I have a general introductory lecture about miniaturization, and then we'll have a specific lecture about the clean room by Lino, about what are the equipment there and how these process equipments can be used to develop processes and create structures in silicon wafers, or in particular, because silicon is the big material for micro and nanotechnology. Then you'll have 2 lectures in the same module, one on microfluidics, so how to make microfluidic channels, and a bit about the properties of microfluidics, so the physical properties that govern the flow at very small scale and dimensions. And then one on soft lithography, which is using this rubber material, so it's the replication of these structures into this rubbery material, and also using them to print on surface and pattern different other materials on surface, so low-cost patterning methods, a bit like printing but at high resolution. And then the module 2, hands-on clean room session. I think that's one of the most exciting parts. Then the lab part, where you're all together in the lab, all together by subgroups, and have different modules where you can explore the different technologies. And then followed by the discussion of brainstorming, which is together with module 3. And then on the last day, the design challenge, different presentation, as well as a very nice and exciting keynote lecture by Dr. Hade Mosseini, who is a material scientist, tissue engineer from Harvard, and who will come and give a lecture on all his different research. Okay, so the outline, are there any questions at this point? Clear? Okay, so I'll give you the outline of the next few minutes, half an hour or so, and I'm going to present you about a little bit about micro and nano. What is this? I mean, so I'm sorry for the ones who are already working in this. It's going to be a bit repetitive maybe, but I hope every one of you can learn something out of this lecture, and well, that's part of the challenge of teaching to an interdisciplinary audience, is that you have different people from very different backgrounds. But okay, we'll discuss about the concept of miniaturization, a little bit about integration and scalability, which is very powerful, which is a concept that have been driving computers, mobile phones, and so it's the integration of different technologies and scaling up so that you can basically increase the number of components and double it every year and have an exponential growth. Then we'll speak a little about patterning. So we have 2 concepts, self-assembly, which we'll touch briefly upon, and then from patterning top-down approaches, where we actually physically pattern, and that's essentially the main concept, the main focus of this course, because that's what we can do in the microfab. And then we'll speak about the original patterning techniques and patterning. And in all these patterning techniques, it's always the creation of the pattern. And then once it's done, then you have different ways of copying and replicating. And usually the creation is really the expensive step or the difficult step. And once that's done, then you can make copies. And then how to make these copies efficient and low cost or high resolution. These are essentially the main topics of, well, which we have to deal with, because that's where we actually come in and work. And then also just briefly introduce the specific techniques that you will use in the course here. Okay, so the vision of micro nanotechnology kind of goes back to what physicists all know this image from Richard Feynman. It's a very famous physicist who had also other interests besides physics. So it was quite a free-floating electron, so to speak. And so he had this vision in the 1959. He gave this lecture where he said, you know, we're doing things macroscopically, but if we could do things small, we could have so much information storage, or could have so many different things at the nanoscale. You can have so many different elements on one little space. And he actually, you know, out of this, he came up with a design challenge where we challenged people to write basically a text on a needle head. And he was hoping that then people would come up with great transformative technologies, and there was a prize for that. And ultimately, someone was able to write something very small on a needlehead, but using actually conventional, traditional technologies. But so this is where the idea is that, you know, nowadays you have basically a USB stick where you have gigabytes of data, and that's the kind of vision that he was proposing back then. And so at that time, of course, this was beyond what people could even imagine, or beyond what people were imagining at that time. And that's the conceptual opening of nanotechnologies. And then why? Well, why would we want to do that? So I think nowadays this is kind of it's kind of obvious. And you can have a high density of information. So from microelectronics, from computer chips, that's what we know, and we're very like that. We can have them portable. We can integrate many functions. And I think, you know, mobile phones are now the examples of having more and more functions. You have now accelerometer here, which can detect how you're moving, GPS, where you are, and of course, in also software integration in terms of data information, but okay, that's even an additional layer that became possible. Then when you're in a biological realm, if we go back a bit more bottom down, it's multiplexing. So when you want to measure many analytes, you want to do diagnostics, we can measure many analytes at the same time. And that's, we have biological technology that can do that, like DNA microarrays or sequencing, where they measure many DNA or sequence many different probes at the same time. And this becomes possible on miniaturization. And I think what one of the big transformations, however, and why nano became so exciting is because we have a lot of new properties that arise. So properties that, when you think of macroscale elements, well, you don't see them, but as the surface starts to dominate, as systems become more confined, there's an example of quantum dots. So quantum dots are small particles that emit light, and these are intrinsic properties of the material, but as the particle becomes smaller and smaller, then they have an effect on the electron levels, and then basically by controlling the size of the particle, you can then, in a simple way, control the wavelengths at which they emit. And so these are some of the properties that emerge out of nanotechnologies. Also, when we think about particles, and that's coming from the physical side, so particles are little specks of particles have been existing for a long time. They were known as colloids. Nowadays, they're called nanoparticles, so there's also a bit of rebranding going on there. But overall, it's of course with a much better control of the dimension that we can then control these properties very strong in a very defined way. And ultimately, what we see, a lot of nanotechnology is essentially an enabling technology. It's not necessarily something you will touch, but it's something that you will find in other devices. And just to give you a brief idea of the length scale we are talking about here. So here are actually two scales, a lot of information on this slide, I'm sorry. But so here, on one, you have, in most cases, you have two length scales here. And so just to put kind of things in perspective, so this is 10.6 nanometers, so that's 1 millimeter here. So you have cells, it's about tens to hundreds of micrometer. Then you have molecules here at the other end of the spectrum, which is just 1 or tenths of a nanometer down here. And you can read the different elements here. And here you have then the different lithographic technique that can be used to structure material at these very small length scales. The same is repeated here, but just for a smaller range of the scale. So we have optical lithography, that's the one we're going to use in this course, which can go down about up to 40, even nowadays up to 30 nanometer, and of course those two very large sizes. Another technique is e-beam lithography, and we'll discuss that later, where you shoot an e-beam to pattern a surface. This is a direct patterning method, it's very slow. Mask deposition, scanning probe techniques, so where you go over the head over a surface and scratch it. And then you have contact printing, so micro contact printing is the one we'll use here too. Here is kind of a approach where you directly print your material on the surface. Here's again the same slide. Here you can have more images from the biological realm for the biologists here. So no light microscopy, I'm sure most of you have used that. Plant, animals, 10 to 100 micrometer, and then you go down to more bacteria, just 1 to 10 micrometer. Phages, so these are viruses that infect bacteria in the 10s to 100 nanometer. Proteins, lipids, atoms. So that's the scale we're working with. And so what is nanotechnology in all of this? So nanotechnology, of course, and also nanoscience in general, it's a bit of a funny definition because, you know, in the end, it's just a science definition. It doesn't mean anything, it just says nano. And nano is just a denomination for a nanometer. And then what does it mean? So there has been a lot of debate about this, and especially in the beginning. Now I think it has been subsided a little bit. But so one of the key aspects of nano is that it has these new properties beyond being small, and so one definition was to say that structures have at least one dimension below 100 nanometer. Of course, that's very easy to achieve because if you absorb, for example, molecules on a surface of proteins or anything, they're just a few nanometers thick. So if you absorb a molecule on a surface, which any biologist will do by doing an assay, well, then you're already having a nanotechnology device. But to make it, and that's obviously not really what people understood with nanotechnology. And so to make it a bit more defined, then they specify that these are designed and made for a process that provides fundamental control of physical and chemical attributes. This idea is that we actually control them, special features of nanotechnologies, and then that they also can be combined to form larger structures. I mean, these are essential elements of nanotechnology. Okay, and so nano revolution, and I think one of the nanotechnology has been with us, according to this definition, actually for many, many years. There's the computer science, so here it's the size of the gates in the microelectronic chips. And this slide, I'm afraid, is already outdated now. Has been going already in 130 nanometer scale in the 2000s, which is at the beginning of nanotechnology, one might think. But since then, your computers have been scaling down, and now you have gate sizes. So Intel, I think it's 30 or even less nanometer, and which is basically what is powering this whole microelectronic revolution. Now we have, but nanotechnology in the beginning was re-envisioned as expanding the possibilities of nanoelectronics, but it has become very much also materials and energy system. And so, for example, now you have new batteries that are coming up where there's nanoparticles inside of them and which enable to have much higher energy storage. But then you have also a lot of hype, and that came out a few years ago. So the Tata Nano or the iPod Nano, which more of you probably know. And so this was the car that was really quite small. Also, what is it, nano? But so that's where it's going. And then on the other hand, nanotechnology, I think, has been with us already for quite some time. And so they did something quite interesting here. They took a candle suit, and then they just separated it in different fractions, different sizes. And then they could see that depending on the fraction size of the candle suit, they had particles which then would emit light at very specific wavelengths, a bit like these quantum dots I was showing you before. So, you know, your car is actually emitting nanoparticles in this way. So that's the kind of, that's where you can see a little bit the rebranding. But so you have this nanotechnology top-down approach where, you know, you very deterministically define features, and then on the other hand, you have small particle size where people now, and I think this, the chemists in particular, and also it's quite sophistication nowadays, can control and synthesize very different particles that have very specific functions. But I think to me the most actually powerful example of nanotechnologies is to some extent us. For us, we have hundreds of 10 to the 14th cells, and each cell inside of it has proteins. And this is kind of a blow-up image from a book, from a good cell. And that's just to give you an idea of the scale that we're talking about. So you have this watch here, and then you have a small silicon MEMS, a microfabricated silicon chip, which is this guy here. And then here they're showing this bacteria, and this has this flagella here, and this is just the output of this flagella. And so this is a nanometer scaled motor. I think the mechanical engineers can appreciate that. And there's a stator and the motor, and this thing rotates at very high speed. It's self-assembled. It's self-repairing, so it fixes itself. And that's just one little part of this whole structure, which is this cell here with two membranes and all proteins that are shuttling it up. And I think that's kind of a very exciting, well, a paradigm of what nanotechnology can do and can achieve. And also, of course, it sets actually a very high bar in terms of what we could or what remains to be done from designers, from artificial design in terms of replicating the functionalities that you might have in the biological cell. And I think that's one of the big outcomes of nanotechnology. We have this convergence of engineering, physics, biology into a realm where all these different forces and different effects interact together, and where we can both try to understand and then use these effects to build new devices that can have new properties and features. Now the main effects, if we go back to the lab, that nanotechnologies might have is that, of course, by miniaturizing, we have a very large surface to volume ratio, and so often what starts to dominate is surface properties as opposed to bulk. So that's, of course, not what we're experiencing in our everyday life, but for people who work with nanotechnology, that's something that you start to learn. And also, of course, you need to control, and that's part of the course. We'll see how we can control a little bit these surface effects to control, for example, the flow of liquid. So the other outcome is that these microscopic fabrication tools don't work. So traditionally, things were done in a model shop, but the model shop for microtechnology and nanotechnology then is what basically becomes the clean room. And so giving shape at microscopic dimensions, there's these two approaches. Conceptually, it's this top-down approach, where you have a patterning and then a pattern transfer onto a surface, and that's what we'll see in detail, and the self-assembly synthesis, which is more like the chemistry approach. And of course, what's very interesting is also to combine these two approaches, where you can have some pattern and then add some self-assembly to add additional sophistication. And so if you look at the self-assembly, that's something that's also you can see in everyday life. So here in the natural world, the snowflake, which is self-growing in a particular way due to the underlying physics of the water molecules and how they assemble and how they go with one another. And so it's always this hexagonal shape. That's the way how snowflakes work based on their physics. Or you have self-organization in biological ways. It's a bit larger scale, where you have the self-patterning of these different discs here in fruit flies, and which is controlled here by genes. So these are different ways of self-assembly. And then you can also try to do that in the lab, and people have been working on that here at the macro scale or the micro scale, where they pattern the surface energy of the surface here with microcontact printing. So it's one of the techniques that we'll learn. And then they throw into a mixture of polymers, which then self-assemble based on their preference in different surfaces. And these kind of techniques, they can be extended down to 10 or 20 nanometer in size. And here we have another case of self-assembly inside of a microscopic channel, or millimeter size kind of mesoscopic channel here, simply by stacking up beads and by controlling the evaporation or the pressure with which the beads are flown in there, you can assemble crystal-like structures of these types of beads. And so we have self-assembly then, as I said, we go also at the nanoscale, and this is an example from nature. So anyone knows what this is? Virus, exactly. Thank you. So this is a virus, and this is then, and it's kind of a schematic representation of vesicles that are in this case assembled and linked together with a DNA linker, where people try, and of course the virus is well known for entering cells and infecting you, and so people try to replicate that in this case for drug delivery, and then they synthesize this type of vesicles. There's DNA linkers here, so they can link different cargo loads here, which then go and fuse with cell membranes and would be carried inside of the cell. So that's the self-assembly side. I'm showing you this. That's a whole world of nanotechnology and nanoscience that people are working on. That's involved in chemistry, and then also applying in nanomedicine. That's not what we're going to cover much in this course. I just want to bring you up to put the rest of the lecture in perspective. So what we're talking about is more than the top-down patterning approaches. Okay, and patterning, we're going back now to the basics of patterning, the direct patterning, so the original pattern formation. And very traditionally, this has been done for a long time. It's very linked, of course, to our culture. And this is the example here. I'm bringing up the Rosetta Stone, which was a stone that had the inscription in 3 different languages, and which allowed the archaeologists to describe basically the hieroglyphs by basically comparing these 3 languages. And so that's a very important archaeological finding. You have mosaics, and of course that we all know, writing. And so here, these are all things where you basically have nothing, and then you start and you create a pattern. And traditionally, I mean, up to the 20th century, patterning was all done for information that was readable at the human eye, which means that it was roughly a millimeter, maybe half a millimeter in size, a bit less, a few hundred micrometers were the smallest size that was actually relevant to pattern. But even here, I mean, these are very slow techniques, and so people, of course, wanted to spread information at a higher speed, and then they invented these higher throughput techniques for doing that. For basically, you have a pattern, and once you have it, you want to replicate it and make many copies of it. And the printing press was one of the transformational inventions in this regard, and has been developed. Where nowadays, you know, every day they print out thousands or tens of thousands or hundreds of thousands of newspapers just from one night in these rotary presses, where everything is assembled in one, or designed and well, basically assembled in just in one evening. And of course, these are very impressive achievements, and it's very important to our society. But again, this is all in the realm of information that's readable by the eye. And these are very mature and established technologies. But then in the 20th century, we come in the area where now we start to develop these micro and nanotechnologies that now can actually go beyond what is seen with the eye, and also require then basically different approaches and different methods for structuring them on the surface. And so the direct micro nano patterning techniques that we'll briefly review here are drop-on-demand and inkjet printers. So this is something probably most of you are familiar with. You can actually go down to a resolution of 10 micrometer here. Then we have electro-discharge micromachining, kind of an etching technique that can etch into metals. Stereolithography and rapid prototyping, so these are techniques that are becoming more and more popular now. And I think you can hear also in the general press about 3D printers. So 3D printers can also be used in the microscale, also with some limitations. But then you have also then from here laser writers, where you can use a laser to directly write on your surface, where you have resolution up to about the wavelengths of light, so 400 nanometer. Then electron beam writers, where you're shooting electrons onto a photosensitive material that then the electrons can break down. And here the resolution is limited by the wavelengths of the electrons, so about 30 nanometer. Or then a focused ion beam, where you have even higher resolution down to 5 nanometer. And finally, the scanning probe lithography techniques, which just briefly overview, where you basically go with a tip and scan it on a surface. And now people have developed the ability of making tips with just one atom at the end of the tip. And with that, they can basically move around. Well, they can both image surfaces with atomic resolution and even subatomic resolution, so they can map electron density, or then they can use that to push around atoms and assemble them one by one. And actually, these were some of the early experiments with STM and AFM that were actually well, both done within IBM, but actually opened up this idea of nanotechnology, where we can now move around atoms and actually structure material almost deterministic way, atom at a time. Okay, now if we go into inkjet, so we have two basic techniques: bubble drop, one where you have two heaters and this evaporates and this shoots a droplet out. So Canon, for example, is one of these develops these techniques. Or you have piezoelectric actuation. So piezoelectric is a material that upon electrical actuation will actually expand or compress, and then by making these lamellas, they apply an electric field, the system compresses, and then this will eject a droplet down of these nozzles here. Now the big advantage is they're very versatile. Then the disadvantage that most of you probably know is that they have a tendency to dry and clog for the ones who have had inkjet and both cartridges. That's how usually they get a bit more money out of you. And well, then they have a lot of applications. They can be used both for patterning polymer, organic or inorganic materials, just chemical materials. So here people use them to pattern transistors made of polymer materials, or you can even nowadays they're being used for actually dispensing cells, and they can even be used for dispensing neurons, and with actually not too much damage to the cells when you actually have your conditions right. So that's one way that now you can hear. And inkjet, of course, it's great because you can pattern something in your computer, and then you press print, and then it will shoot and create your pattern in a very rapid way, or in a relatively rapid way. So the other technique for patterning, the second one that is a direct patterning technique, is E-beam lithography. I just told you before, so E-beam, you first will coat a wafer with a very thin layer of resist. So what's commonly used is PMMA, so plexiglass, for the ones who know, that's one of the very popular resists. And this is coated in a layer maybe 10 or 40 nanometers thick, and then it's exposed to an e-beam, which is a scanning head, which shoots electrons, and can basically then break the bonds of the PMMA, and this can then be developed using a developer, so something that dissolves the PMMA, but not the ones that haven't been exposed. Then this is being covered with a metal. This is a lift-off technique here, where then the resist is being completely dissolved, and so the pattern is then left on the surface. And this pattern, for example, can be used in here to etch into the material with different etching techniques. You'll see in the next lecture. Or vice versa, or this can be used in electrical contacts, for example. The big advantage here is very high resolution, so up down to 30 nanometer, but it's very slow. It's a serial writing, so if you want to write a millimeter with a 30 nanometer line, even when it's writing fast, it's actually a very slow technique. So when you have large areas, millimeter square, a millimeter square is a large area for an e-beam. And something that's even higher resolution is the focused ion beam. So here, instead of shooting electrons, it's actually delivering ions directly, and these ions basically then they can bombard the surface, and they can be focused in a very narrow spot down to 5 nanometers. And then by tuning the chemical conditions of your chamber, you can either have the ions deposit on the surface, so they accumulate there and they write, or by changing the pressure, you can have them etch away the surface, and so make very narrow little holes in here. So this is a 500 nanometer scale bar. Again, here high resolution, control over etching and deposition, so this can be very interesting for making very small holes. Again, same issues, very low throughput, slow setup, vacuum, very expensive instrument. And one of these instruments costs probably over $1 million. Then scanning probe lithography, a very brief overview of that. And here I'm showing more the biological applications of it. So this is the dip pen lithography, where now they use a tip, which is not as sharp, maybe not a single atom, but which is then basically coated by a liquid film. And in this liquid film, you can have very different types of molecules. I mean, there's instruments available here at UCAM, I think. You can coat it with organics, or silanes, or sials, that molecules that self-assemble on the surface, or proteins, or DNAs, or particles. And by writing them, you can deposit these molecules and pattern your surface. And one of the challenges here is that it's only one ink at a time. So you can have in theory many tips together, but typically they're all coated with just the same ink, or if they're coated with different inks, then you don't really have full addressability. And so multicolor is still something that's really challenging. It's quite versatile in terms of you can take different things one after the other, but then again the throughput is relatively slow. So these are all these original patterning techniques that I just wanted to briefly show you. And as you can see, for all of them, essentially, they are just slow. I mean, it's great, you have a great freedom in what you want to pattern, but it will take you a long time to make a large area. And so, like for writing and printing press, people have then developed these techniques to replicate. So that once you have created a pattern, you have then ways of copying and making it many times. And so the first we're going to briefly show is photolithography and microfabrication. So that's kind of the content of this course. How you can pattern photoresist in 2 and 3D. And then we'll briefly overview the different techniques that kind of are a bit, that go from there, where we have molding, that's the one you're going to do in the lab here, it's kind of simple to implement, and hot embossing and injection molding, which are commercial techniques that are used for making mass-produced products. And then finally, soft lithography, where we can then use these soft stamps to print different inks, viscous inks or molecular or solid inks, so we can transfer particles. And then just the last thing on mold fabrication, where you can make these molds for, which can be used for all these different techniques. Okay, so photolithography and microfabrication, that's the module 1 of the course. And the way this works is that you'll spin coat a layer of photoresist, in this case SU8, it's an EPON, it's the same that's used, the same type of chemistry that's used for tissue slice embeddings, for the ones who are into that, which is then patterned on the surface. And then we use a photomask, so you'll see Lino will talk about that in more details, where you have openings where light is shown through, so it's collimated light. And wherever, in this case, this is a negative resist, so wherever the light goes in the resist, it will cross-link it. And then once it's cross-linked, well, once this is exposed, you'll take this and put it in a bath and etch and expose it. And this bath basically will then reveal the pattern. And this can be used, for example, as a mask for further etching. So you can then use it and go into a dry etching machine, a gaseous etching machine, which can transfer the pattern into the structure, or it can be used, as we saw before, for depositing a metal. And then, for example, you can dissolve the resist and create metal structures on the surface.
What's the mask made of?
So the mask is it's a glass mask, and then they have a chrome pattern, a very thin chrome layer, and that's enrolled either in contact. I mean, so it's so it's going in contact or by projecting lithography. So it's using a big glass plate. Maybe Lino, you have one mask here. Okay, yeah, here. So you have one here. So that's a mask. Lino will hand them around. And so when you want to do that, you ask, and you design it on your computer, and then you send it to a company, and they will create the mask for you. And then you get it back, and then you will use it to make your pattern inside of the 2D.
And the resolution of the pores?
So, for example, here in our fab, in this case, you can go down to about 1 micrometer. But that then becomes hard. I mean, even 1 micrometer, these long lines, you have to be good in the fab. So there's also this sort of practical aspects of it. So, for example, but if you think about microelectronics, for example, Intel is using these photomasks. So they have a projection lithography, so they have lenses in between and so on. But they go down to 30 nanometer. So that's what's possible nowadays. So for us, it's about 1 micrometer. That's what you could do here. Okay, and then once you have a structure, it's great, but then you can replicate it further. So with 1 mask, of course, you can make many wafers, right? You have a mask, you expose, and you make. But still, you need to go to the clean room. Then if you want to be even, depending on your application, it might be enough to actually replicate them into rubbery materials, so PDMS, and you will see that later too. PDMS is like a bit of jelly, and so you just pour it over. You can take your wafer, you can pour it over, and you put it in the oven. It cures, and then you peel it off and trim it. And so for the ones who have been baking cakes in a shape, it's the same idea. You have a shape, you put it in the oven. Here it's just 60 degrees. If you want to cure it well, it's 24 hours, and then you can take it off, and it keeps your shape. And this is kind of this gooey material. So it's very simple, and you can do structures also down to a micrometer without problem, even lower. You can do down to a few hundred nanometer. Now, this is very popular in academic labs, and that's the one we'll see. And the reason it's very simple to implement, but of course, for commercial production, this has not been very successful, because each time you want to make a copy, you have to immobilize your master, your structure here for several hours. So of course, you can't make that many copies in high throughput.
Sorry, David.
Yes.
Can you sterilize the PDMS? Can you put it in an autoclave?
Yes. Autoclave, UV, ethanol, everything. Yes, so we use it to print proteins, but people use it in combination with cells. So there's one risk, is that there's always uncross-linked material in there. The cross-linking is never complete, and so that can slowly leach out. And so for example, with neurons, you have to be very careful when you do that. Yes?
So probably working with viscous fluids, usually bubbles inside that. So is there any way to like eliminate the bubbles in the PDMS?
We're going to the details now. Well, there's many ways. Yes, the design, treating the surface, pre-filling with ethanol, putting your device first in a vacuum, so it then becomes, because this oxygen can get dissolved here, right? So for example, people, they put it in a vacuum for a long time, then they put the liquid, and then this sucks up the air that fills in. So there's different approaches, yeah. So it's always an issue. I mean, it's not always. Actually, with the type of microfluidics, by self-filling microfluidic, it's usually not an issue. Some others it's an issue. Yes?
What techniques are used to fabricate the mask itself?
The mask itself, I think it's a laser writer, typically. So it's I think it's a photoresist system too, where they write with the laser and then they develop it, right? Or
yeah, e-beam lithography also.
E-beam for high resolution. Yeah.
Direct laser writing, the resolution is 0.4 micron. Okay, and with e-beam lithography, we can reach 10 nanometer.
Okay, thanks. Okay, any other questions? Okay, so once we have these soft stamps, and you'll see they have a property of being self-sticking onto surfaces, but we can use them either as stamps for inking and printing, so like when you're a kid, you had these potato stamps you were doing, except here, usually we don't print viscous ink, we print actually molecules that absorb a monolayer of molecules, so to speak, which you can transfer. Or you have the option of sealing it first on a surface and making little channels and then filling your channels like we were just discussing with different types of chemicals. That's two things that you will do in the course here. And now, just a brief overview of different other techniques that are used actually more commercially to make structures, but which are slightly derived from these. So one of them is hot embossing, where you would have a polymer like a thermoplastic, so plexiglass or polycarbonate, and then you heat up here your structure, which again you could do by microfabrication. When you press it together, it will fill the mold, and then this will cool down, and as it's cooled down, you would separate it. So here it's very high resolution. Still, it's a bit slow because as it doesn't take several hours here, it still takes like several minutes to heat up and cool down your system. And so in commercial terms, this isn't being that useful. These are the type of machines that were used for that. That's very nice. You can do resolution structures here. And Steve Chu from Princeton was pushing this like a few years ago. Already have like something like 10 nanometer posts and this very high aspect ratio made by these type of techniques. Now the real winner in terms of commercial application is injection molding. And so here it's the same than before. In fact, the difference is here now you have you don't need to heat your system here. This is cold. And what is being here is your pre-polymer. The polymer is actually being melted. And then at very high pressure, it's being injected into the mold. And almost instantaneously, it then freezes down and becomes hard, which means you can then open your mold and take it out. And so this is, for example, used for making CDs and DVDs. And there the production rate can be 1 or 2 per second. So it's like, so it's big machines that inject, freezes, opens up, and then closes, injects, and so on. And that's how then you can have mass production do that. Now this is widely used commercially. Unfortunately, the machine to do that and the mold to make a mold costs it's very expensive. The mold is very expensive. So the entry barrier is quite high, and so that's why you rarely find this type of instruments really in academic setting. I mean, I see there's one chemical engineering here, but it's not for really microscale applications, more for macroscale applications. Okay, and these are then the kind of machines that do that, and that's the type of structure they can do. Also, not only CDs. So CDs are like features down, or DVDs down to 300 nanometer in size here, and these are structures. These are much larger structures than in this case. Okay, and so coming to the end of this lecture, and so what I'm just bringing up here Moore's law. I mean, kind of a bit underlying theme of this lecture here. So Moore's law is this idea that you have a doubling of the processing power of computer. So now Moore's law, we call it Moore's law, but actually Moore didn't formulate it as a law. He was, if you go back and read some of the original text, he was making an observation very early on that they were able to double actually the processing power of Intel of their computers every 18 months or so. And this continued for a while, but then the semiconducting industry, they actually took that as this idea of doubling their processing power, they took that as their targets. And the reason for that is if you're making elements for computer chips or for computers, of course you have to, if you're making a hard disk drive for a computer or a new processor, you want to make it cost effective and you want to make it also novel enough. But so if you're making too advanced, then it's going to be very great, but it's going to be too expensive, and so no one will want to buy it. And conversely, if you're actually making it very cheap but actually not powerful enough, then again you will be out of sync. And so that to make sure that all the different people involved in this could actually have some knowledge about what's going to be the next generation of devices, they basically then said every 18 months we want to kind of double the processing power so that people knew what kind of specifications they might have to design for. And so to some extent it's a bit a self-fulfilling prophecy because I want to double it every year, and then yes, it's doubling every year. And so that's of course that's in the sense of it maybe less surprising. On the other hand, the fact that over 40 years they have been doubling, and I'm sorry, this is kind of getting out of range here, have been able to double essentially the processing power. Of course, that's something very impressive, because as you can see, this is an exponential scale here, and of course, this has had all these transformative effects, where now you have mobile phones that are more powerful than computers that have been just a few years ago. And these transformative effects are also happening out in the biological world. So we have technologies which now have adopted these micro technologies for biology. and one that has been the first one that has taken that was DNA microarrays. They started in '94 with a chip with 400 spots, and by using actually photolithography to pattern the chips in a company called Affymetrix, they were then able to double on a very rapid pace, actually faster than the computers, the number of elements on their chip. And they reached 6.4 million, and then they stopped. So they hit the wall. And so for the ones who are in computer science, computers are always afraid of hitting the wall too. And I mean, they hit the wall to some extent because maybe there was no demand, but also because another technology came along, which is sequencing. And so sequencing, there's of course a lot of biochemistry going on, but a very important part of sequencing is actually microfluidics. So the way things are shuttled in sequencers are with microfluidics, so they're a small part of that. But, so this, again, you can see this micro nanotechnology is actually a very important part in sequencing. So nowadays, was in the 90s, 80s, 90s, there was the Human Genome Project, which was a multi-billion-dollar effort over 15 years. And so now we speak, I think in 1 year from now or so, we'll be able to sequence a human genome for less than $1,000. Which, you know, is like regular other tests, which means in a few years from now, we can all get sequenced if we want. And we probably all will, because it's just going to be something you just can do. And okay, that's what we're working on. But so just the conclusion for this part, this kind of big overview. So a lot of micro nanotechnologies have emerged in the last 15 years with a lot of transformational changes. Two fundamental concepts. You have to create a pattern, that's the hard part. And it's done expensive in clean room, and you can do it replication. So part of it is also done in the clean room, but you can take some of that replication outside, actually take it into your lab and do high resolution structures inside of your lab. And so micro nanotechnologies have revolutionized computers, biology and medicine, and I think that's only the beginning. And so here you can all become part of it by learning and applying it. And so before I let you go, just because this is a course, you know, you're invested. And so a little bit homework. As I said, some of you have some ideas for the brainstorming sessions. I challenge you all to actually come up with some ideas for the brainstorming sessions that we can discuss there. It can be very exciting if you have a good idea, and can be very beneficial to you. So it's a bit, you know, you'll get out of it what you're putting into it. So if you have ideas and you bring them up, and we'll discuss them, I'm sure you'll get something out of it. So if you like, select a device or structure related to your project or not, that you'd like to fabricate using micro nanofabrication processes, and think about it. You prepare some sketches on how you would fabricate it, how you would use, so you'll see, Lena will show you microfabrication, how you could use the processes that are available in the microfab to try to build it. And part of that is actually to make a process flow. So when you speak in the microfab, and I'm sure Lena will talk about you a little bit, is you have to think in terms of process flow. First I make a photolithography, then I etch it, then I make a second photolithography, and then we make the schematics where we have the cross section of our wafers like I showed you. The first, then exposure, then you transfer the pattern. And that's the way how we have to think when we use this when we use this microfabrication. So that's one of the skills that hopefully you can develop a little bit during this course. And then we can discuss it during the brainstorming session. So that's the first one. And then after that, we'll have the design challenge. I'll have a presentation here. And so again, if you have been thinking about for your own brainstorming, that will help you for the design challenge. There is prizes also with the design challenge. And it's going to be actually quite a complex challenge this year, but I'm sure we'll have some very exciting solutions coming out of that from this group. And with that, I want to thank all our sponsors, which are helping make this actually possible. So we have the ISS and Abbott, which are our major sponsors this year. Then we have DICE, Nano Quebec, and CMC Microsystems. So CMC, and we'll come and talk a little bit here too about what they're doing. So CMC is kind of they can build things for you if you use some of their processes. So they're kind of they're used for Canadians. They actually collect all the orders so that you can access some technological processes which are not accessible otherwise if you come as an individual researcher. And then we have also support for the wafers from VRS Materials, so that the wafers you will be using have been donated, the neuroengineering training program, Microtools Nanofabs, which is giving us access to the preferential, giving us some support, and also with Lino is participating here, my lab, and then the Postgraduate Student Society, who is also supporting this. And with that, I'm going to thank you for attention. Happy to answer any additional questions. Yes.
Is there any FIB focus ion beam around in the industrial?
Yeah, Polytechnic they have a focused ion beam. So we don't have one at McGill.
But actually it's not working.
Oh, I don't know. But not working now, you mean, or not in general?
No. I don't know, it had problems like for the past year, so that it was like today, and for the whole month they repair it, so you don't know what it's.
Yeah. So yeah, you have to see. These are big, difficult machines, so they break down. Yeah. Yes.
Are there any of these processes that can integrate with stuff that's already at the nanoscale? Like, say, you already have a cell, you want to print a link to that cell. So you have to be able to see that cell and then.
Well, so yeah. So there's, for example, people they polymerize proteins in solution with cells. So they can do that to some extent. Or there is actually here, this building in physics, there's people who use AFM to poke cells and deliver things between cells. But okay, I mean, once I didn't maybe mention many of the microfab clean room things are very harsh for biological systems, right? So they typically done separately. Then you have these soft lithography techniques with the polymer stamps, which today you can interface with cells, but usually you print and then you fill your stuff, or you grow them later on. Then if you want to direct act with your cells, well then, okay, photonic materials, microfluidics, so people culture them in microfluidics and deliver different chemicals by switching the flows or locally delivering things on the cells. So in a fluidic way, or then with a laser, or then with AFM if you want to be at a smaller scale. So in this sense, yes, there's different tools, but and each of them has advantages and limitations. Okay. There is no more questions. We move to our next speaker.